About
Yeah. Yeah, it's still doing that white screen, huh?
Responsible for moving something.
Y'all right. People everywhere. We'll come back over here. All right, appreciate it.
thank you. We'll try again tomorrow.
Point, because I never would have thought to look at, and something flashes. Yeah, so.
Thank you all for coming. I appreciate you all being here on a Thursday afternoon to celebrate the work that our students have done over the summer. They are going to, in just a moment, present to you in brief the progress that they have made over the summer. Just to give you a sneak peek of what's happened. They will also, in the spring of this coming year, 2025, be asked to share the research in a more formal way. So while this is not a formal presentation, they will be giving one either at a conference or at a publication or in some kind of way. So be looking forward to that. My name is Hillary Richardson. I don't know everybody in this room, but it's good to see some familiar and unfamiliar faces. I'm the coordinator of undergraduate research. And I'm going to tell you a little bit about the program that the students participated in. They had to apply with an application letter from a mentor and a full page proposal on what they wanted to do during an independent study. And this year was the first year we were competitive. So the people that you see here are a really great group, and I'm not saying that just picture in front of me. You guys have done some really good work this summer, and I'm proud to say I think this is one of the strongest cohorts we've had since this program has started. So after they submitted the letter and the undergraduate research advisory committee reviewed them and said these look great, they embarked on either a 5 or a 10-week independent research program, and that's 45 hours of work per credit hour. So basically a full-time job. So they've done a lot of work. We're here just to celebrate it. The program also came with a little bit of funding, so students received some funding if they needed supplies, and they're here to kind of tell you what they did. You see a cycling of the students, but they are Moira Cairns, who's in the back, music therapy major, Kane Petty, biology major, Zareen Raya, another biology major, Anna Sandlin, a history major, Jessica Schramek, another history major, and Ethan Wilkins, another biology major. They're going to present just briefly what they've done, and there will be time for questions at the end if you have. So, are y'all ready? Are you guys ready? We're going to go alphabetical. Okay, where are we going to kick us off?
Questions at the end of each presentation. Very end, yeah. I've got you if you want. There's a clicker in there.
Good afternoon, everyone. This past 10 weeks, I've been privileged to be able to extend a research project that began at New York University by analyzing 28 pre-existing interview transcripts of autistic adults, describing their experiences of music in everyday life. Before we begin, I would just like to give you the definition of autism as per the American Psychiatric Association. This will help in better understanding the emerging results that I go through later. So the definition of autism is the persistent impairment in reciprocal social communication and interaction, as well as restricted, repetitive patterns of behavior, interests, or activities. And there is also an age of onset and pervasiveness in life.
So this topic holds personal value for me, as there are many people in my life, friends and family, that have autism. I also have worked in the field of special education and also in supportive roles for nearly 20 years with people with autism, and my goal was always to enrich their lives. It was my use of music in these roles and seeing the effect that it had in these roles that really influenced me to go back to school and to earn my Bachelor of Music with an emphasis in music therapy and become a music therapist. During my clinical hours, I had a particularly impactful experience while working with a client with autism. He was a young boy who rarely made any sort of vocalization. So that was one of our goals, was for him to make a consonant sound or a vowel sound of any kind. After 8 sessions, the reason that the picture of the kibasa is up here, he particularly liked ambient instruments. And so we were rolling the kibasa on his arms, up and down his arms, up and down his legs. And I was singing, roll the kibasa, you can feel it on your arm. And so we went around, and later on in the session, he went over and took the kibasa and started to roll it in his hand. So I went over and sat with him and I sang, roll the kibasa, you can feel it on your hand. And I just sang it over and over. And about 3 times in, he sang, hand, right when I was going to sing hand. So I sang it about 12 times, and every time I stopped the song, you can feel it on your, he sang, hand, on tune, like in tune. And it was just such an amazing experience for him to be communicating with me that way. So this area of music and autism holds a lot of value for me personally. It's only been recently that researchers have begun to interview autistic adults, autistic individuals, to inform the research that will eventually impact those individuals' future supports and others with autism's future supports and interventions. So what we are doing will highlight the thoughts and feelings of those in the autistic community, which is really important for future music therapy and client interactions. So this past spring, I took a course as part of my music therapy major, music therapy research. And during that time, we looked at the general sociology of music as well as music and autism. Just kind of looked at the general research and wrote a literature review on what was currently out there in the research piece. In looking at how we would analyze the data that we had from this project at NYU, we chose to go with thematic analysis because it does the best job, we felt, in highlighting the voices of those being interviewed. These are the initial codes that we started with. You can see on the left that there's each one with music, and then on the right, each one without music. And so we're coding for both to see trends and patterns. So music and emotion would be defined as, we would define it as music is linked in some way to the subject's emotion, whether it means evoking emotions or sustaining emotions, changing emotions, exploring emotions, understanding music as an expression of emotion, that sort of thing. Music and self-awareness is when music is linked to the person's awareness of self, whether it's their identity and internal self or their body through exploring movement, dance, and that sort of thing. And connection, general sensory motor refinement and connection. Music and aesthetic is when the interviewee shows an interest in the music based on its own value. So whether it is on its own properties and how they relate to each other. So it could be on the organizing principles of music, whether it be the meter or the rhythms, maybe the chord structure, this sort of thing. Part-whole relationships and just interest in the beauty of the music itself. Music and social would be any time music is linked to the social life of the subject. This could involve the act of making music with others or going to a live music performance, understanding relationships through music and etc. Music and supplemental is when music is considered a secondary or accompanying activity to another activity, such as listening to music while driving or while doing chores around the house, that sort of thing. So initially, when we were coding, I used a pen and paper, I did it manually, and then we moved on to the in vivo software. So I'm going to show you just a short video on how I was coding. So you can see at the top, so this is not one of our interviews because for confidentiality purposes, this is actually an interview that I pulled off online with Dolly Parton. She did in 2001. And you can see at the top, it has the interviewer. And so I don't want to code anything that the interviewer says, obviously only what the interviewee says. So, so I just go and I highlight what fits under the code. So here she's talking about a lot of the types of music that she listened to growing up. So I coded it under genre. But then this one little section here, she's talking about making music with her mother. So I coded that under music and social. And then this last piece, it says, a lot of old gospel songs were written to sound old, like I say, by a lot of my own people. So I put that under music and social, or sorry, music and self-awareness, as far as identity. She's identifying herself with this group of people. And then I have another little piece here. It says all of us, but she's referring to what the interviewer said. I put that under identity, music and self-awareness identity. But I added an annotation there to put it into context so that when we pull up the data under the particular code, we know what it's referring to. So I identifies as musical within the family unit. So that's just a little snippet of how I would code. Now, when I first started coding, I did a lot of the annotations and started to see trends within those initial codes. And that's when the themes kind of began to emerge, new themes and subsets of themes. These are the 6 phases of thematic analysis. Right now, we are in phases 3, 4, and 5. And although this looks quite linear, it really is circular when it gets to phases 3, 4, and 5, because we're searching for themes, we're finding those themes, we're refining and defining those themes, and then more themes come up. And we have to make the decisions of, do we split this theme? Do we merge these themes together, and so on and so forth. And so we're kind of in this phase with, or in these 3 phases, until we really feel like what we have as far as themes go really reflects well on the data that we've collected. So this is where we are right now as far as themes. So you can see now on this map, I don't have the codes without music. I'm just focusing on the music codes right here. You can see how there have, so these would be called the parent codes, and then we have child codes that come off of them, and then another set of child codes off of them. So as we go through, now we're coding for all of these different themes and pulling information into these subsets of themes. And some of them we may end up paring down. We may not end up using one like genre, for example, if it doesn't really yield information that we're interested in. But for now, we wait and see, because we really want the data to speak to us as opposed to us looking for what we're looking for. Because the point is to have the voices of the autistic community highlighted and have what they're saying to come out of the data as opposed to what we're looking for. So I spoke a little bit about pairing. cutting out themes later on and merging and splitting themes, and this is a whole process that takes a while. To really kind of get to a point where we feel like the themes reflect the data. And so you can see here, music and aesthetic has paired off into, not sort of paired, but split off into 4 child codes. We've got, and then within that, we've got this one down here, familiarity, sorry, I can use this. Familiar, there it is, familiarity and predictability. And so this came up as this came up repeatedly, which is of interest because it's part of the diagnosis of autism, is restricted repetitive patterns of behavior, interests, or activities. And so then we saw some things come off of here, and these could be future subjects of inquiry. So for example, creating music and the music environment. So we saw a lot of things come up with composition, improvisation, as far as creating music, or also being the leader, like being the lead band member, being the lead role in performance, these sorts of things. And so looking at, so we've got lots of different life paths and choices and things, but you've got this pattern emerging, so that can really help to inform future inquiries. As we get kind of closer to the end of our going through phases 3, 4, and 5, we can do something called data mining or data mining, which is the use of quantitative methods through the in vivo software or other software like it to discover trends that may have been missed in the coding process. This is the top words, most frequently used words under the everything that I coded for aesthetic. It's no surprise that music is right there in the middle, and a lot of these words kind of in the middle wouldn't, you would expect them to be used quite a bit. But when you get into the edges here, you've got words that were used at least 20 to 30 times. So you've got some interesting ones like dense, or improvising, composition, and then you've got one up here, colors. And so it's interesting to kind of see on the edges, this is still pretty frequently used. And so it might be something that we want to look at further, kind of dive into further and see why are these things so frequently used. But there are some cautions for using data mining. These are tools to draw attention, but not to draw conclusions. So it can really, maybe one interviewee said the word dense 25 times because they particularly like that word. So it's something to kind of go back into the data and look and examine how much do we need to look into this further. But it's a great tool to be able to see these things. So what's next? This spring 2025, we are hoping to do a presentation at the Southeastern Region American Music Therapy Association Annual Conference in Birmingham. And we also hope to produce a report for publication on the findings and possible new areas of inquiry. Thank you so much.
Remember which one of these is you, King?
So, my name is Kane. I worked with Dr. Hagee on the effects of formaldehyde respiration on lizard toe shape. We used 3 different lizards. We used one gecko, one phelsuma, and 2 anoles. This one in the middle is your phosphema, and these two on the edges are urinals, for like, visualization purposes. What are topaz? It's the lizard meat, but, to get more into that, it's the sticky parts on a gecko's or a nose toes. It allows them to stick to the rocks and trees they live on. They, just kind of evolved that way because they needed a way to climb. They have these tiny hair-like structures. So this is your toe, and then those are these folds. And if you look very carefully, there's a bunch of little tiny hairs. For reference as to how tiny, that is a red blood cell. Like, they are extremely small. They're called setae. There's a lot of different ways you can say it. That's preferably how I like to say it. But it allows for friction and like molecular forces to create bonds with surfaces. And it just, there's a bunch of different types. But you don't have just like one shape. You have all these little funky ones, especially this one down here. But lizards are kind of just all over the place when it comes to the shape of their toes. The reason we're interested in specifically formaldehyde is because formaldehyde is commonly used to preserve reptiles, fish, and amphibians. There's a bunch of different formaldehyde preservation techniques, but for this one, we're looking at formaldehyde. What formaldehyde does is create these cross links by adding extra bonds that allow specimens to keep shape and to prevent bacterial decay. And we think that shrinkage would be occurring because of these bonds, specifically with width compared to length. And I'll explain why in a second. And we're interested in the shrinkage because it will determine whether or not studies that use preserved specimens need to account for said shrinkage in their research. This is, this is studies that we are kind of referencing for our stuff. So just kind of give like a visualization as to width versus length. That line kind of shows where the bone would be in your toe pads. This sort of visualize like that's bone right there, and then surrounding it is like these soft tissues. And down here it kind of shows that like, there's different types of bones and toes, and it's kind of cool. But we think that based off of these studies that we were previously looking at, that width would change compared to length. We primarily looked at 3 different studies. 2 used lizards, 1 used a fish. The one that used one of the ones that was a lizard study, they took x-rays to take their measurements of different limb lengths as compared to what we did and what the second lizard study did, which we took external measurements. So they're looking at measurements that are extremely like point A on a bone to point B on a bone. We, in the second study, are looking at like bone measurement, but also like the stuff in between. This study that used x-rays also used green bonds, which are significantly larger. So we looked at it, just to kind of, it's lizards. But our second one, the second lizard study, they, 1, used external measurements the same way we did, and 2, also used anoles, which is one of the ones we used. So it was used kind of as a predictor. This is their results in the middle. So you can see that like sections like, head length, femur length, tibial length are lengths that like, it's a bunch of bone. So you don't see a significant decrease, and you kind of just see it kind of do its little thing. But their results showed in lengths where there is a lot of tissue, there is decrease. You wouldn't think spine would do that, but there is soft tissues in between the vertebrae of the spine. So, when there's shrinkage, even though it might not be like a whole bunch, it's still there. So we use this study to kind of predict our results and gather our hypothesis of thinking we're also going to see shrinkage. And then our third study that we looked at used fish. They didn't see, like, they showed some change, but no overall change in shape. Meaning like no overall pattern or shrinkage, but they did use geometric morphometrics, which is what we use, which I'll explain what that is in a second. Kind of moving on to what we did instead of background, in 2018, 32 specimens, which there are more that we got, but we used 32 for this one, were collected in Hawaii, pictured, measured, and then euthanized and put in formaldehyde to sit until 2023, where they were taken out of their formaldehyde, measured and pictured again. We took specifically, there were pictures of their rear foot, and we're specifically looking at the fourth toe on that rear foot, to kind of keep our data very consistent between 2018 and 2023. We used geometric morphometrics type like programs. Primarily we used 2 programs and then 1 coding software. So we used TPS Stitch, which this is kind of what you'd be seeing, very much a lot more colors. But TPS Stitch measured XY coordinates to create these little dots, which are called landmarks. And then we used another software called ImageJ, which collected area. So it would look kind of like that. That area that's like outlined in white is what the numbers we want to get. And then we combined those 2 and used R, which is a coding software, to create plots to visualize the change that we're thinking we're going to see. What we've gotten so far is that there is change, but statistically, it's not enough to rule out change. So, and you're going to bear with me on this plot. It looks a little confusing, I know. This this shows like the wider to thinner topaz, and then this is like a change in like overall shape. These light gray right here are 2018 measurements, and these are 2023, and then you have your legend of the different ones. There are differences because they're not exactly on top of each other, but they're still extremely overlapped. And they also go in random directions. There's not like a trend that's like, oh, we're going this way. So, we're like, okay, there's difference. What is it? Like, can we negate the difference? We did some statistical works to see if species in year affect shape. And that's where we got that it's different, but not enough of a difference. This is kind of an easier way to see it. So again, it's the darker line is 2023, the lighter gray one is 2018. So you can very much see like different right there, but they're still extremely on top of each other. So right now it's looking at, we're so far seeing results that are consistent with, it's different, but we don't have to be worried about it when doing research. And there's been some past projects in the lab that did work with topaz. So that supports that work, and it also supports future work concerning topaz. We're looking at putting this in and putting it all together to publish this. And then this is going to be further presented at a conference next year. Thanks. Thanks. I didn't learn you once.
Hi, I'm Zareen, and today I'll be telling you a little bit about the project that I've been working on with Dr. Burns. So my project topic is genome analysis of a bacteria called Aeromonas hydrophila, and I'm focusing specifically on a strain or variant called S14452. So Aeromonas hydrophila is an animal pathogen. What that means is that it affects different groups of animals, like reptiles, amphibians, fish, mammals, and birds, and it includes humans. It can also cause an infection called motile aeromonas septicemia, or MAS, in catfish. And this basically causes rapid death in catfish, and as you can see, like, the catfish over there, it's dead because of like internal bleeding and then organ damage and fluid accumulation. The reason I bring catfish up is because the bacteria that I've been working on was isolated from catfish, and that catfish was infected with MAS. So a little bit of background on this project is that in 2009, this bacteria actually caused an MAS epidemic on catfish farms in southeastern US, mainly in Mississippi and Alabama. And this introduced like hypervirulent or more infectious strains of this bacteria. The one that I'm working on, S14452, is also one of those like newly sort of introduced strains from the epidemic. Over there, you can see like a phylogeny where these are all like the new strains, and this has never been sequenced before, so that's kind of why I decided to analyze it. Yeah. So this is an overview of my methodology. There were two parts to how this was done. There was a wet lab part, and then there was the bioinformatics pipeline that I followed. So we isolated the DNA from the bacteria and sent it to a lab to get the sequencing done, where the DNA was basically broken down and fragmented so that we could like interpret the bases that were in the DNA. And that was done using like Illumina software. And then I put the bases through quality check using a software called FastQC, just to make sure that all the sequences and all the bases were good to go for assembly and annotation. And because those are the two main things that I worked on, I'll be talking about those more the rest of the presentation. So I did assembly with a software called Spades, and then I did another version of that using Bowtie 2, and then annotation was done using rapid annotations using subsystem technology software. So what is assembly exactly? Assembly is basically, remember the fragmented pieces of DNA that I talked about during sequencing, is basically putting those together. So the fragmented pieces of DNA are basically called reads, and those are what I got back in the file. And I uploaded it to Spades software, and it has this inbuilt algorithm that allows it to find overlaps in the DNA bases. And those overlaps are then put together to form larger fragments called contigs. And I got back 218 contigs. So just for like your understanding, it's not the whole like DNA sequence of this bacteria. We're still figuring out how to make that happen. To do that, we would have to connect all the contigs and find the sequence that it's in. So to help with that, there is like a previous version of like another variant of this bacteria that has already been completely sequenced. So I took the 218 contigs and I aligned them to this reference strain, which is ATCC 7966. And all these blue and red parts that you see here are the bases that match the reference genome. But then you can see like the highlighted bits, and those are like single nucleotides have been like changed. So there are more of these because I got to use the browser, so I got to like scroll through millions of base pairs, and there were like a lot of them, but you just get to see those two. Yeah. And to figure out the differences between like the reference and also just annotate like what type of genes were there, I moved on to like annotation. These are the general annotation results that I got. And I found out that the genome that I'm working with comprises of 5 million base pairs, a little over that. And for your reference, the reference genome is actually 4.7 million base pairs, so it's pretty close. And I'm thinking that if we can kind of like put the contigs together and form the full genome sequence, it's gonna like come to that number. And it has a G+C content of 60.8%. This is relevant because G+C content indicates the stability of the DNA and like of the bacteria itself. So a higher G+C content corresponds to higher stability. And it also has a total of 5,013 predicted genes, of which 4,903 are coding sequences, which means that these are the genes that actually like produce a protein, and those proteins have actual functions that help the bacteria like survive and also infect hosts. It's also got 110 RNAs. So these are what help with protein synthesis. And because I'm more interested in how it sort of like carries out the infections and affects hosts, I looked into virulence factors. Those are the things that help it infect hosts. So it possesses type 1, type 2, or 4 secretion systems, and these are essential for a pathogen like Aeromonas to be able to infect and invade hosts, sorry. And over here, you can see that my annotation results basically categorize the genes into various different functions. So you've got like genes for virulence, you've got genes for carbohydrate metabolism, and genes for like amino acid synthesis and stuff like that. And then here, I looked into the metabolic pathways as well, just because I wanted to understand like how it infects all these different types of animals, even though like the animals have different environments internally. So because of like the number of metabolic pathways that it has, it shows that it can survive in various different environments and therefore affect a lot of different like, groups of organisms. And then I did a comparison-based annotation. So over here, I went back to the reference genome and I aligned it with the one that I'm working on. And I found that the orange bits over here, these are like differences in the proteins. And then I looked into it further and I found the genes that I wanted to look at were to do with the virulence. So I found 25 unique genes, and not all of them correspond to virulence, but I found 5 genes for like celiac acid metabolism, 1 gene for type 4 pilus, and then 1 for multi-drug resistant efflux. And what that means is all of these genes correspond to like all of these proteins, and I found like 3D diagrams for these. So all of them help the pathogen infect the host, evade the host immune system, and also fight like any kind of antibiotics that the host might take. So yeah. And the limitations of my research is that most of it was done computationally, and all of these softwares that I used are prone to error. So that's a limitation. And then many of the genes could not be annotated by the software that I used, which is RAS, because they code for hypothetical proteins. What that means is that these proteins don't actually have like functions that we know of. So we don't really know what those genes could be doing. And another thing is that because all of this is like sort of acting as like a first step to do experiments in wet lab, we don't actually know the true effects of these genes unless we actually knock out some of the genes and do it in like wet lab. So for future research, that's basically what I plan to do. I plan to investigate whether these genes significantly contribute to the virulence and like do what I'm thinking it's gonna do. And the second part of it is I would like to compare the genome analysis that I've done to other Aeromonas species. So back to the phylogeny that I showed you earlier and compare it to like other more infectious strains of this bacteria. And another thing is like the next step is to make a publication out of it, like most of the other projects here. So, and this was funded by Mississippi INBRE. Here are my references. Thank you for listening.
Ready. New project or, okay.
Good evening, everyone. I am Anna Sandlin, and I am a senior history major. So my summer scholars project was an in-depth research project on Emma Odie Poole, who was the director of physical education at Mississippi University for 47 years. Dr. Kicker, who was my mentor, and I were very excited to start learning about Poole's life and to create a written biography that would highlight her significance on the college and her students. So this is my process of where I started. I immersed myself into Emma Odie Poole's papers and the Beulah Culbertson Archives and Special Collections at the library here at Indigo. I spent 3 hours a day for the first 2 weeks combing through each of the Poole papers. I would take pictures of the files and I would upload them onto a Google Drive. And then I provided 2 pictures of some of the examples of like there were newspaper articles and a lot of letters, and she had a lot of papers that she just like wrote stuff on. So that is one of her better handwritings, but it was also a slow process in trying to read her handwriting because she didn't always write to be read. So it was difficult to sort through those. But I then organized all the information that I had in my like box and folder. And then I created the database on Zotero so that I could have specific papers easier to access and to cite. So once I had all the information from the archives, or had enough, I started writing. And within the span of another 2 weeks, I had written a 20-page research paper. So I can't give you all the information I found, but I will give you some. So, after high school, Emma Odie Poole studied at the University of Missouri, the University of Illinois, the Chautauqua School of Physical Education, the Chicago Normal School, and a few other small colleges. However, it's very unique and interesting. She never received a college degree because she only took the classes that she wanted to take. In 1907, Henry Whitfield, who was the president of the INC, invited Poole to organize the physical education department, and in 1908, Poole took the position as head of the department. Emma Poole instituted many traditions at the school, such as the May Day Festival, the Zoo Off Drill, plays such as the Joan of Arc play, and the Junior Freshman Wedding. She also organized Taft Day, the Pageant of Columbus, and the school's 50th anniversary mass. Even after she retired, she still dedicated her time to the school. I included this picture, and it's of the Zoo Off Drill, which is one of the traditions that Poole dedicated a significant amount of her time to. The drill is important to her and her importance because it instilled a significant amount of discipline, and it allowed her to teach her students that they were capable of more rigorous activities. So to further expound on Poole's importance to the college and her students, I had to analyze the idea of Southern white womanhood through the 20th century. Southern white womanhood has a distinct history, and many state legislators believe that white women's higher education and institutions should have been implemented to make better wives and mothers, ultimately conforming to the gender ideology at the time. However, women students and faculty did not always conform to the ideas of male legislators or school administrators. White women's campuses held the possibility of being more than finishing schools for women or making them 20th century examples of the antebellum Southern Belle. When the INC was established, many administrators and legislators attempted to subjugate the women. From its inception, the college saw a strange mix of intentions and aspirations, with goals to educate Mississippi women for possible employment, to educate women in professions deemed suitable for white women, and to ensure that women on campus remained under the authority of Southern men. But the women demonstrated their commitment to forging the college experience that suited them. So, Pauline Van de Graaff Orr, who was chair of the English department at the INC from 1885 to 1913, presented a feminist voice on campus and challenged directly male administrators and legislators She was extremely radical. On the other hand, Emma Odie Poole did not directly challenge male administrators or publicly break with them over their decisions regarding the school. But her example and teaching radically transformed women's lives, and what they believed that they could do. Her goal was to inspire her students to see that they were capable of more than they had previously ever even dreamed. And how with nothing more than their bodies, they could defy expectations. Even though both Poole and Pauline Vandergraaf Orr were suffragists, Poole stands between the more radical activism of Orr and acquiescence to gender hierarchy because her life and leadership presented the middle way, one that recognized the limits of her authority and sought permission from male administrators, but also by demonstrating an unflinching commitment to excellence and by pushing her students to see themselves as capable of what they set their minds to. Her method was empowerment through discipline and games. So, for my future directions, in the fall, I'm going to be completing my capstone project for my history degree. And so I will continue to develop my paper by connecting Poole's impact on her students more and to further expound on the educator of Southern womanhood and how women fought the gender hierarchy in Southern colleges. I plan to participate in the UW Undergraduate Research Conference. And then I would also like to submit my paper for possible journal publication in the Journal of the City. I also want to pursue an advanced degree in history, and I think that this project is a good stepping stone in that direction, and it'll provide me a potential writing sample and then a future master's project. I've provided some select references, and I just wanted to say thank you all. I'm so grateful for this experience. But I also helped Salem, Sweet, and Bridget at the CHC, along with Mona Van Velde at the Phillips Park Archive. Most of my time at the CAC in the conference room where I took boxes and went through each and every one of them. It's quite a mess. The filing cabinet was in the stairwell. That's where a majority of the boxes were stored. The Salem had told me to store the boxes against the far end wall of the room and put them piled up. I just paid to live. These photos were evaluation of the Concert Council around the and they found them in the pamphlets and brochures box. They had gotten misplaced after I got to leave. I searched for them before I got here in June, but I'm very glad that I found them. It's really interesting. These were some of the photos that I found in another box, which were pre-renovation of the Rosensweig Art Center. And as I found out that the stairwell that's right here, that when you see when you walk into the building, that was made during the renovation. So it threw me for quite a loop when I found that this photo had no stairwell. But I also found more. The timeline of the Arts Council is that in 1979, the Arts Council received a room at the Columbus Auditorium, according to the article. In August of 1985, they moved into an office of the Princess Theater. Sometime in 1996, they moved into the Tennessee Williams Welcome Center, and that was mentioned in this last little sentence
of an article in the August showcase. And then in 1998, the Rosenzweig Arts Center was donated to the Arts Council. And in 2001, it opened after renovations on September 6th. And the sources of it, these two came from the archives, which was the Commercial Dispatch from October 1998. These were the August 1985 showcase, and then this was August 19th, 1979 Clarion Ledger, where I got first information from. Now these were some of the logo contests in 2012, so these could have potentially been the logo that we see today at the Columbus Arts Council. Personally, I think that one looks a little like Star Trek, and then that one looks a little like Monsters Inc., but they didn't choose them. But what I've learned is that I had to train myself back to categorizing every single item because I really wanted to, which I think is a good sign that archive work is probably going to be the way I go. I really enjoyed going to a boxes despite all the nuts and I sometimes used to avoid getting a paper cut, but I feel a bit more assured that this is the kind of career path that I want to go towards, so that helps reassure me. And old paper smells very distinctive. As for future plans, is that during the course of my process, that I kept track of all the list of alums that I came across, and I'm hoping to cap some. Also, there's the $10 need for the Rosenzweig Arts Center.
All right, last but not least, Ethan, are you ready?
My name is Ethan Wilkins. I'm going to be a junior biology major here at the W. And my project was I picked up on a project that we're about to finish. My mentor, Dr. Hedda, he gave it to me. And it's a project that he developed for undergraduate students to start learning many of the biochemical molecular processes to learn in the lab to use on future projects. So the project specifically was trying to figure out the most efficient, effective way to detect endogenous CFTR species in different cell lines. And I'm going to go through all the jargon and go through all that. So I'm going to talk about today the approaches and techniques that I use over this summer scholarship program, and also some of the findings that we found. So some terms and definitions to give a background. So what we do is we work with cystic fibrosis. And cystic fibrosis is a genetic disease that is caused by a mutation to the CFTR protein gene found in the CFTR protein that is coded, which is the protein that is created from the CF gene, is found on the surface of epithelial cells. And epithelial cells can be found from lungs to the pancreas to the digestive system, and all of these different organ systems are affected during cystic fibrosis. So CFTR, it stands for cystic fibrosis transmembrane conductance regulator. I would not say that. It is a plasma membrane protein found on epithelial cells that acts as a chloride ion channel, which this channel affects the electrical balance of the cell. So the term endogenous means is a cell or cell line that is produced naturally within an organism. So it's naturally how it is naturally occurring within you and I or any other sort of organism. So exogenous is a cell or cell line that is produced, it is produced artificially outside. So what it is, say, and we're going to go through different types of cell lines here that we're going to talk about. So what happens with an exogenous cell or a cell line is you take the cell from an organism and then you are genetically manipulating it to show the signal that you want to make it easier to work with and other things like that. So one of the cell lines that we used was called CFBE41O-. So this is a non-transfected mother cell line from the human cystic fibrosis bronchioepithelial cells. So this came from a human that had cystic fibrosis, and this is what we use as our negative control. because it shows low amounts and it's been tested to show low amounts of that residual CFTR protein. So our CFB wild type is this same mother cell line that is genetically manipulated to show a higher cystic fibrosis protein sequence. So this would be our negative control, which shows very low amounts of residual cystic fibrosis protein. And this would be our positive control because it's actually been genetically manipulated to show an upregulation of this protein. So that would be kind of our positive and negative control for our experiments. Next will be CIFBAC. This cell line actually came from a human pancreas. The person had pancreatic cancer and they did a biopsy and we were able to gather some of these cells. So, and this person had cystic fibrosis and we were using this because this other cell line, which is CAPN, also from a human pancreatic, this patient had pancreatic cancer, except for, so CIFBAC, this person had cystic fibrosis. CAPN, this person did not have cystic fibrosis. So that's how we're going to tell the difference. And they were not genetically manipulated in any way, shape, or form. So a little bit about CFTR. So like I mentioned, CFTR is a plasma membrane protein that acts as a chloride ion channel. And mutation to this protein causes it to misfold and allow it to not be properly transported to the plasma membrane to function as it's supposed to, which causes that differences in regulation, eventually causes the mucus buildup that is commonly found in the respiratory system and digestive problems and other things like that, which causes cystic fibrosis. So this is just a brief, quick and dirty overview of just protein synthesis. So you're inside of the nucleus of a cell, you have all your DNA and everything like that. And then that goes through transcription to create a single-stranded RNA. And then that goes through translation through ribosomes and everything else. And then it becomes a sequence of amino acids to become a protein. So that's just a quick and dirty way of what we're going to talk about next. So one of the techniques that I use was called RT-PCR. So RT-PCR, the first part, RT, actually stands for reverse transcriptase. So what we do is we remove the RNA from these cell lines and we go through the RT process and we're actually able to turn the RT or the RNA into a complementary DNA strand. So we're actually doing the reverse part of this right here. We're turning RNA back into DNA. And then the PCR stands for polymerase chain reaction. So we're able to take this new complementary DNA and amplify it so that we're able to detect different expression levels and other things like that for specific sequences that we're looking at. So qPCR kind of goes hand in hand with that. So qPCR also uses this polymerase chain reaction to amplify these sequences. But the difference is we're able to, as it says, quantify and measure and have different numerical values to be able to analyze these different expression levels, and we're able to measure them and be able to compare between our negative control and baseline and cell line to cell line. So there's two types of qPCR. There's dye-based, and what dye-based does, and it's the older version when this process first came out, and for the longest time it was state of the art, and then how it's used. And what it is it takes the dye and it implants it onto a certain primer that attaches and goes through this process, and you're able to measure the amount of amplification by the amount of fluorescence that it shows. The second is called probe-based qPCR, and this is what we use for our data. And the reason that this is so much better is the problem with dye-based qPCR is the probability that you have non-specific pairing. So what it does is it pairs and binds to a sequence that you don't really want it to, that's not your target sequence, and it causes a skew and abnormality in your results. So probe-based qPCR allows for more specificity, and you're also able to probe different sequences within a single sample. So we're able to look at different sequences of this DNA at one time instead of having to do one for our endogenous control and one for our CFTR. We're able to put it all into one well, and we're able to run the sequence and everything show what we would like to. So here, this is actually one of our diagrams that we created. So our A, again, is our positive control. So green line is our CFDR, and that's our target protein. Actin acts as our control to just show consistently from cell line to cell line that the concentrations, everything that it stays consistent. So line A shows our CFDR for our positive control, as I talked about. B is for our negative control. And these two lines in the middle show those differences in both of the both of the CAPN1 and the CIF-back cell. And something that we noticed consistently through our results is that CAPN had more CFDR expression than CIF-back. And that's kind of one of our findings that we will go into toward the end of this presentation. And one of the benefits of qPCR is having the ability to have RQ values, and we have cycle thresholds and other things like that. So we're able to look at the differences between, as here it says, 16-fold higher expression for CAPN1 and 6-fold for CIF-back. And we're not able to have that information if we don't use a qPCR. Next, we're kind of an old technique. It's called gel electrophoresis. And what happens is you load these samples onto a gel, and you use electricity because these proteins have positive and negative charges, and the protein will transfer through the gel and it will be arranged by size. So you have the larger protein at the top, smaller proteins at the bottom, and you are able to differentiate between those different sizes. So this is actually a DNA gel, so it's kind of the same situation. And here it's very faint to see, but so we have our negative control, which shows this is our CFDR region and our actin region. And this supports what we found in our qPCR, and it shows no signal for our negative, a lot of signal, or relative, but a lot of signal for our positive control, very little for our CIF-back, and it's kind of fuzzy, but slightly more for our CAPN, and then pretty consistent with our actin. So that just goes another technique that we use to support our findings from our qPCR. So this is actually what's happening inside of the cell. And when we use techniques for cystic fibrosis, they show up in 3 distinct bands. Band C is a mature, completely fully glycosylated form that is found on the plasma membrane, has normal function, everything like that. And then band B and A are immature, so they are usually mutated, and they get stuck within the cell and they cannot perform the normal function, and which causes cystic fibrosis. So they show up as 3 distinct bands. You have C, which is at mature because it's higher up on that gel, like I talked about. It's going to be heavier, it's going to have a larger size due to the post-translational modifications. And then B and A are very similar. They're very similar in molecular weight, so they usually show up as 1 distinct line, and they usually go together. So next, we're going to use we use a technique called Western blotting. I did not specifically do this. This data was done by a previous student, but I will definitely go along with this. But this technique use electrophoresis, and then take a membrane, and then you transfer that over to the membrane, and you expose this membrane to different antibodies that has tags, and you're able to measure the tags that were grabbed onto your target protein, and then measure how much of that protein was actually there. And we can do that from well to well, so we do it from sample to sample. And this shows the difficulty of using this technique for these endogenous cells. So here on the left, we are using these exogenous cells that are genetically manipulated to show this higher sequence. And we actually use, it's 50 micrograms of protein used here with a very strong B band and C band for these 2 cell lines. This one is actually a normal cell line. This one is the defective mutated version. Here we use 500 micrograms of protein sample to even have a comparable sequence result. And you can see that just from the amount of protein just by our actin, of how hard it is to work with these endogenous cells, because it requires so much protein and so much antibody, which is very expensive. So it's not a very efficient or effective way to sequence these cell lines. So findings is that, as I talked about, RT-qPCR emerges the most convenient method for detecting this endogenous CFDR expression. This biochemical methods are shown to be not very cost effective due to the cost of antibodies and having to use so much antibody and then using much more sample to achieve not very great results. So we found that qPCR was the most effective way to do this. So the detection of this method holds promise for cystic fibrosis diagnosis because using these endogenous cell lines is the same as a patient coming in with cystic fibrosis or a child being born with cystic fibrosis, and their cells are endogenous. They're not genetically manipulated at all, but you're able to use these cells, and having a deeper knowledge of how to work with these cells can help with the furtherance of cystic fibrosis research. Another finding was due to the low amounts of residual CFTR, as we showed in the DNA gel and the qPCR, the CIFBAC cell line has the potential as a parental cell line for transfection, the same as the CFB 41O-, that can be used to be used as another cell line to be genetically manipulated and used in the furthering of cystic fibrosis research. Any questions? That's fine. I just added it.
Thank you all for coming, and we'll take just a few minutes while we set up our refreshments that I promised you, and we'll be delivering. To see if anybody has any questions. I also encourage you to ask these students questions as you mingle with them one-on-one, but does anybody have any questions they want to ask right now? Can we give them all one more hand for doing? And if you are joining us via streaming and want to type a question, we'll ask it for you as well. Streaming or
What's that? Military tanks or like a water? Yes. Well, so it's portal, so each of your fingertips is going to be the size of the piece of paper. So it's scaled for the